Let's start with this afternoon's lecture. I'm Jimmy Hsia. I'm from University of Illinois Urbana-Champaign. Let's take a quick survey of the students. How many of you are engineering students? Vast majority. Biology students? Thank you. Medical students? One? Sort of. Okay. And graduate students? Yeah?
Global enterprise for mechanical and molecular
Close. Good. Good. Okay. GEM4 stands for Global Enterprise for Micromechanics and Molecular Medicine. Micromechanics, molecular medicine. So it's M to the fourth power. Okay? And we actually, can you close? Thank you. It was established back in 2005. One of the major activities that GEM4 organizes is a summer school series. Okay? And this is a list of summer schools we have organized so far. Okay? All the way starting in 2006 at MIT and till 2012 over here at MIT and also at Imperial College London. I shouldn't put the question mark over there because they have a theme. And for each of the summer schools, we have a particular theme. There are several different very special characteristics or very special features for GEM4 summer schools. One is for each summer school, there's a theme. Starting from the first one focused on infectious disease, and this one focused on developmental biology. Okay? And the second feature, unique feature for GEM4 summer schools, is that we always have hands-on labs. So usually it's lecture in the morning and hands-on labs in the afternoon. Okay? The feedback we've got so far is most of the students, or all of them actually, enjoy the hands-on labs tremendously. Okay? If you go to some other summer schools, very often you go into the lecture room and you listen to the lecture. That's about it. And we started with the concept that without students going to do these things, to work on these lab modules, to make things happen themselves, their learning experience is limited. So that's why we started this. Several of these, for example, the first two were organized at MIT and National University of Singapore. Then from the third one on, we got a grant from National Science Foundation. So the 5-year summer school series from 2008 until 2012 is supported by the National Science Foundation. Starting from Caltech, focused on cardiovascular systems. 2009 at University of Illinois, focused on enabling technology and so on. 2010 was interesting. Actually, there's a history behind that as well. We wrote a proposal. I'm the PI of the proposal to NSF, proposing that we have 5 summer school, GEM for summer schools. And the first one we proposed was at Caltech. The second one was at Cambridge University, UK. Third one was at University of Illinois. Fourth one at National University of Singapore. Fifth one at MIT. So it's US, Europe, US, Asia, US. Proposal was ranked #1 on the panel. And they all said that we should support this. But the program director came back to me saying that I gave you 2 options. One, I support 3-year summer schools, Caltech, University of Illinois, MIT. Second option is I can support the 5-year series, but you have to find 2 additional US institutions geographically, strategically located in the US. So because of that, Texas Austin and Georgia Tech were added to that series. It turns out 2010, we had this summer school at, not summer school, we have a World Congress of Biomechanics in Singapore during the summer. So because of that, NUS organized a GEM for summer school. Plus, Texas is too hot during the summer, so we decided to have a winter school that was actually in January. Okay. So that worked out perfectly. And this is actually the last year for that NSF support. And I was talking to a student, asking the student's experience. We're planning to resubmit the proposal to renew the summer school series. And perhaps what we can, if you have had a very good experiences with the summer school, what we can ask you to do is to help us, maybe provide some testimony saying that, oh, this is great, and this is a great opportunity to train students and so on. So that would strengthen our proposal. Okay, and you have an opportunity to do that. At the end of the summer school, we have evaluation. So provide some comments and suggestions and whatever you think about the summer school. Okay. Actually, the first GEM 4 alumni Facebook was established in 2009, way before Facebook became popular. Okay. I heard that you established 2012 Facebook group. Congratulations, and I hope you use it to your own advantage. That I don't know about this particular school, but the one at Illinois, this was 2009. We actually had the couple, husband and wife, participating in GEM 4 summer school together. Do we have one over here? You do. All right. Okay. Congratulations. So I was talking to a friend saying that, we have a GEM 4 couple. We have a husband and wife participating in GEM 4 summer school. So she said that, oh, that's wonderful. So we have GEM 4 couples group or whatever you call it. And then I said, no, it's not GEM 4 couples. It's GEM for couples. Okay, we'll give you some GEM to Okay, and GEM4 was established in 2005, as I mentioned, by Professor Subra Suresh. He was Dean of Engineering at MIT. After that, he was nominated by President Obama to become director of NSF. He's now director of NSF. He's been director for a couple of years now. And since 2010 was the fifth anniversary of GEM4, so we actually made a little movie.
Pleasure to welcome you to the launch of Global Enterprise.
This is Subra Suresh.
And molecular medicine, now commonly referred to as GEM4.
GEM4 is a fundamental.
This is the former president of MIT.
And the program for this launch
Just stepped down this year.
When you look up the literature, you see all the 56 papers in Chinese which have been translated to English, all working together from these various universities and cities.
Could we actually create brand new tissues in the body? And this is a really, I think, very powerful example of interdisciplinary research.
Since it's the fifth anniversary, we call this movie GEM4 at 5.
GEM4 was created in 2005 as a way to bring together faculty, students, researchers from many different institutions around the world to have a common interest in addressing major challenges related to human health. The superhuman idea of a virtual institute that will draw from different disciplines around the world to look at some of the most exciting problems that lie at the intersections of science, engineering, medicine, and public health.
We're creating a new generation of engineers/scientists.
This is somebody you all know.
Part of moving biology forward is to help it become
Bob Nary is another associate director of our center, EBIX.
biology, as opposed to focus at simply the molecular and cellular level.
The collaborative effort between engineering and biology would have huge impact on things like healthcare, environment, security.
You can think about cell biology, nanotechnology, imaging.
Biologists realize that they too now have to know something about mechanics. Just as a person coming from mechanics already realizes that they need to know something about biology if they're going to try to contribute to this field.
We really need to have a team. It's not just to do experiments, we need to model it into computation. So for any single lab,
Another person you know, sitting right there.
But science is biological, and the solutions, many of them are engineering in nature.
Part of the mission of GEM4 is to create new educational opportunities and educational content by bringing together experts from very different fields and very different geographical locations to come and teach in a program for which there are no existing textbooks. And the people who participate in summer schools are the world's leading experts working at the very cutting edge of technology. These are 2-week-long courses. Very intense. All these students that are about to come together on a single campus and rotate some other campuses. In addition to the lectures, the students have had the opportunity to get hands-on experience in labs in different institutions around the world.
There are places all over the world that are asking us if they can have a GEM4 summer school because they realize that it's a way to really bring in the biomechanics community to their country.
We have major revolutions in many different fields that have previously not overlapped. One of the initial projects was in the area of malaria. A number of institutions got together and tried to formulate collaborative platforms through which different aspects of malaria could be addressed.
In cardiovascular research, a very critical issue is the development of plants. And therefore, with our effort within GEM4 to understand how the mechanical signal due to the change of collateral patterns induces black formation. And of course that leads to a better understanding of the cardiovascular diseases. For the next 5 years, we will go beyond just biomechanics. We will include synthetic biology issues related to cellular machines.
Promote interdisciplinary research to break down the walls between the different academic units, because that's where the action is.
Seek to develop new ways in which we can understand human diseases, and we can try to develop, if not a cure, these methodologies to address human diseases on a global scale.
Just in case you don't know, the picture in the person in the middle is sitting right there. Do you recognize yourself, Takami? Yeah. By the way, one of the Gen 4 summer schools we were planning to organize was in Japan in last year, actually 2011, at Tohoku University. Actually, Professor Takami Yamaguchi was going to organize that. Unfortunately, the earthquake came and the tsunami destroyed many of the infrastructures over there, so we were not able to do that. But we're so glad that Professor Yamaguchi continues to be actively involved in this. Thank you very much, and sending many of the Japanese students over. So we actually circulated after the earthquake among our friends, and many people sent emails to Professor Yamaguchi with their concerns. Another thing probably Professor Roger Kam mentioned, certainly Professor Gangbao mentioned, this is the Science and Technology Center we have, called Emergent Behaviors of Integrated Cellular Systems. And the goal of that is to build biological machines using multiple cell types. And one important component is an international component, or international collaboration. Gen 4 is one of the major international partners for this. So you are now the second cousin of this NSF Center. The center is about 5 years for $25 million and renewable for up to 10 years. So it's essentially a $50 million investment from NSF. Okay? So these are all activities that are very much relevant to what you are learning for these 2 weeks. And hopefully you'll be pursuing. I saw many more engineers than biologists. And it's the essence of this organization is biology, engineering, medical science collaboration. Okay, so take advantage of this, meet your counterpart, and start collaboration. Okay, the thing I'm going to talk about it's relatively simple. I'll try to make the case that nanotechnology is important in biological research. I'll tell you two short stories. One is about cell responses to surface nanoscale features. Okay, and the second story is using micro-nanotechnology to measure cell property. Okay. Cells are sensitive to its mechanical environment. This is not new. You probably have learned many of these things. One of the pioneer works in this area was done by Dennis Fisher and his co-workers. And they started to differentiate stem cells on substrate of different stiffness. And the reason, the hypothesis was stem cell differentiation may be governed or controlled or manipulated to a certain degree by its environment. We know that the environment in the brain is actually the mechanical property, or the stiffness, elastic modulus, is very low. It's on the order of 1 kilopascal or so, or a couple kilopascals. Whereas the muscle, the stiffness is on the order of 10 of kilopascal. Bone, maybe 50, 60, near 100 kilopascal. Okay. So because of these things, Dennis Disher actually put the stem cells on different substrate and indeed observed that if the substrate is very soft, then the stem cells would differentiate into neurons. In the intermediate stage or range, stem cells would differentiate into muscle type of cells, and at higher stiffness level, stem cell would differentiate into bones. So this is something that that was sort of eye-opening to biologists at the time, and became a major story. Okay? Now we also know that cells would respond to surface features. Okay? If you think about this, you have cells that are attached to some extracellular matrix environment. Okay, all these environment, no matter how smooth they look, there's some roughness on it. Right, the surface of the table is actually very rough under the microscope. Okay? And Johann Schwarz at Max Planck Institute actually came up with this idea. He had the technology of putting gold particles, nanoparticles, on silicon substrate. And he can make them with different spacing. For example, in this case, it's 28 nanometer spacing, 58 nanometer, 73, and so on. Okay? Then what he did was he put the cell on different substrate and see how they respond. For example, this is a substrate with gold nanoparticles of roughly about 73 nanometer apart. These are, I believe, fibroblasts, and they actually would behave relatively normally. So they stretch, they migrate, they move, and so on. However, when he put the same cells on the substrate with gold particles of 58 nanometer apart, the cell just pancake out. Very different behavior. Okay? So obviously the cells can sense its mechanical environment very clearly. Okay? And there have been many people, this is also the Max Planck group, who are looking at cell responses to surface grooves. And these are large, relatively large-scale grooves. And there's this is also quite large, 10 micrometer grooves. Okay? And these grooves are made by lithography, or it's the same technology that you make microfluidic devices. Okay? And what they observe is cells would line up with the groove direction. Okay? Now the question is, what exactly happens? Okay, what do the cells sense? And there was one hypothesis. This is by Mike Sheets and Viola Vogel. They were saying that very often these cells would sense the curvature as one geometrical parameter or factor influencing the cell behavior. Sheets actually took a movie of a fibroblast pulling on a collagen fiber, which is quite amazing. You perhaps don't see it very clearly in the back, but if you look at that, you can almost see that the cell has a sort of a hands over hands pulling action. it's amazing a single cell as a machinery can do all these things. Okay? How does the cell sense that there's some structure, and how does it reach out and apply force on this? Okay? Now, if the curvature is the controlling parameter, what we started to ask, the question that we asked was, indeed, whether curvature is the controlling parameter, or maybe it's related to something related to groove height or step height of these features, or maybe some other parameters related to so-called feature size. What is this feature size? We don't really know at the time, okay? But it must be geometrical. Okay? And what is the mechanism that the cells use to sense these different feature size? That is also something that we are not quite clear. Okay? So what we did was we started to look at ways of generating different feature size systematically. Okay? And this is one way, one method we use to generate feature size. It's actually a very simple way to do that. You can see that for this particular case, this is an AFM image of this. For this particular case, the wavelength of this waviness is about 2 micron. Okay? And the amplitude of the waviness is about 180 nanometers. And this is an optical image of that surface. And the way we did that was we have a PDMS piece. Okay? Then we stretch the piece mechanically, maybe 10%, 15%, 30%, whatever we do. Then after under stretched condition, we oxidize the surface, so you form a very thin layer, couple of nanometer or several nanometer thick layer of oxidized layer. Okay, the oxidized layer has a significantly higher stiffness than the PDMS substrate. Then you let go, relax it. Because of this mismatch between the oxidized layer of stiffness and the substrate, you form this well-defined sinusoidal waviness. So depending on the oxidation time and stretching strain, you can actually control the wavelength as well as the amplitude. Okay? Not always independently, but somewhat you can control that. Okay, so that's what we did, and so we can generate a number of different surfaces with different wavelength, amplitude, and the curvature turns out to be proportional to amplitude over wavelength squared. Then we started to look at that. For example, you don't see it too well. This is wavelength of 1.2 micron, amplitude of about 140 nanometer. The cells don't align at all with the feature, but for 1.65 micron wavelength, amplitude, even smaller amplitude, cells clearly align themselves well. So there seems to be a cutoff feature size. In this case, it appears to be wavelength that controls the whether the cells would align with the feature or not. Okay? And we didn't see any clear dependence on curvature, though. We actually tried to correlate the alignment with the curvature. That didn't work too well. Until we started to look into some other, so what we decided to do was to look at the really what happens during the alignment process. Okay? So we took some video of these cell division process or proliferation process and try to see whether the alignment has some pattern in it. So this is a particular video. The wavelength is actually relatively small. It's about 800 nanometers. And if you look at the, this is the from day 0 to day 2, so 48 hours. Okay, these are C2C12 cells, which is a mouse skeletal muscle cells. And there's a pattern, there's actually underlying pattern in the vertical direction, but they seem to be relatively random. Okay, they don't seem to align well, which is consistent with our picture, right? Our photograph showed that if the wavelength is too small, they don't align. Okay. However, okay, let me also explain something else. Using this as an example, you see the bright spots once in a while in this picture, in this video, right? These are the cells that are about to divide. Look at this bright spot. Okay, some of these, this is just divided. Some of, okay. Okay, there are two processes. One is cell division is very often associated with these cells balling up. So right before they divide, and adherent cells would tend to ball up like this. When they ball up like this, you see a bright spot. Okay. However, if you do that, if you continue that, the same experiment from our 48 hours to 96 hours, or day 2 until day 4, end of day 4, you see a different picture. You see that the cells are proliferating and they're becoming more and more confluent. Okay? As they become more and more confluent, there seems to be a pattern emerging. In fact, when they become almost completely confluent, they all line up with the features, even for the small feature size. They all line up vertically. Okay? That seems to indicate that there are two competing processes. Okay? One of them is the cell interaction with the substrate. In this case, if the substrate feature size is now not quite defined yet. If the substrate feature size is small, cells don't really sense the feature size at the very beginning. Another process is cell-cell interaction. Okay? Cells themselves would interact with each other. When they are sparsely distributed, their interaction is such that they would orient it somewhat randomly. But when they are becoming more and more crowded, they tend to align themselves. Okay? So it's these two competing interactions that eventually gave rise to this phenomenon. It should be pointed out that the cell-cell interaction and the cell matrix interaction are very different interactions. Okay, cell matrix interaction, the mechanism, molecular mechanisms are very different. Okay, so we're collaborating with Professor Peter Wang, who gave a lecture last week, and studying potential molecular mechanisms during that process. Okay. Now the question is why we're studying this. Remember, EBIC's goal, or at least one of the major goals of EBIC, is to build biological machines. And in order to build biological machines, you need to be able to manipulate cellular behavior. And one of the things, for example, for muscle cells in our body are all lined up. But if you just use culture dish to grow cells, they orient it very differently. So they don't have the bundled strength of a muscle cell. So this is maybe one way we can manipulate cell behavior, or through the understanding of that. Okay. Now another short story is this using micro-nanotechnology to measure cell properties. Certainly, this is not. This is something that people have been doing for a number of years. Maybe one of the early work using micro-nanotechnology to measure cell property was pioneered by Chris Chin, who's also lecturing. He used micropillars to measure cell forces, and to generate this force map. Another work well known in the community is by C.T. Lim and Subra Suresh. They are actually using optical tweezers. These are micro beads and glass beads. They can use laser to control these beads, and then using these beads as what's so-called optical tweezers to pull the cells to measure the cell stiffness. Okay? In this case, they are interested in malaria-infected cells. Okay? And this is the cell cycle of malaria infection from a healthy cell on this side to fully infected cells. Okay? And they show that a healthy red blood cell, these are red blood cells, they show that a healthy red blood cell is actually very compliant, or you can stretch it relatively easily. Out of power? Over here. Okay, and on the other hand, heavily infected cells, malaria infected cells, become very stiff. So with the same force, you can stretch a healthy red blood cell to a huge deformation, but for an infected cell, you can barely deform it at all. It happens that all these red blood cells, or majority of them, would actually circulate in the body and go through these capillaries. When they go through these capillaries, they undergo huge amount of deformation. You can imagine that if the cells can no longer deform, it would strongly affect how these cells would function. Okay? And the way one of the things that we did was using micro-nanotechnology to measure cell mass and cell growth. This was published in 2010 in PNAS. One of the central questions in biology about cell growth is, okay, if you look at the cell growth process or tissue mass growth process, there are essentially two different processes. One is cell itself may grow, okay? It becomes larger, bigger, fatter. In fact, the fat cells, they don't divide. The fat cells over here, okay? The number of fat cells is more or less fixed since early on, maybe 3 or 4 years of age. I don't know the exact age, but very early on. That's why childhood obesity is so important. Okay? Now what happens when you drink a lot of beer is each of these cells would grow. Okay? Grow to a certain degree that the beer belly is become that big. Okay? And one of the ways to deal with that is to take these cells out by suction. A second mechanism that tissues grow is cell proliferation. In other words, increase in number of cells. Okay? And this is the growth cycle, how the cells would grow and divide and become two daughter cells. Okay? Typical mitosis cycle. So there are two different hypotheses. One is that the cell growth is controlled, or the rate of that is controlled by the nutrient getting into the cell. Okay. If that's the case, you can imagine that the nutrient diffusion into the cell is proportional to the cell surface area. So as the cell grows, the surface area is larger, and there's more channels for the nutrient to diffuse into it for it to grow. Okay. You set up a differential equation. It tells you that if that's the case, then cell growth would be power law dependence on cell mass. Okay. Another hypothesis is cell growth and proliferation is actually dependent or proportional to the number of DNA duplication and production and the protein production. If that's the case, you can actually think about the case that it's almost like the cancer cell production. The more cancer cell you have, the faster they grow in number. Okay. You set up a differential equation that gives you exponential growth curve. Okay? So the question is, which one is the case? Nobody knows. The reason nobody knows is nobody has a very accurate way of measuring cell mass as a function of time. Okay. One of the methods that people use to measure cell mass as a function of time is this cantilever beam method, where you have a little beam, and for cantilever beam, a thin beam, there's a resonance frequency. So if you plug it, it vibrates with its resonance frequency. Okay, that's a mechanical property of the beam. Okay? And you can actually look at the frequency, measure the resonance frequency relatively accurately. However, you can imagine that if the mass of the beam changes with an added mass, for example, then the resonance frequency would shift. By measuring the resonance frequency shift, you can determine how much more mass you have added to the cantilever beam. Okay? So this is the basic principle that people use to measure cell mass in the past, at least. Okay. These are some pictures that Rashid Bashar at Illinois and co-workers, they have studied that and used that technique to do this. The problem with that is oops. Let's see. Something's wrong. Let me close the program. By the way, Roger, we were able to play the video with the audio. It's even worse. I know what to do. Never happened before. Let's see what happened.
It must be this room, Jimmy. We had somebody else whose computer just stopped in the middle of the presentation.
Really?
Yeah. We restarted.
Okay. Let me skip that particular page. See whether this, okay, this one works. Okay, so the problem with that particular technique is these cells are, or many of them are migrating cells. So they would move on the surface of the cantilever beam. And we did some brief estimate. If an added mass moves from the tip of the cantilever to somewhere near the middle of the cantilever, then the error you have is about 100%. So the method is essentially useless. Imagine that you have a measuring technique that would consistently give you something around 100% error. Okay? So this is the problem that we want to solve with the new technique that we developed, which is a technique that involves micro-technology to develop a sensor, a platform like this. Okay? So this is a picture of that little platform, of MEMS platform that we developed. And we did some finite element simulation, which showed that if you have a mass, added mass, anywhere on this particular little spring, little platform, the error of the mass measurement by measuring the frequency shift, resonance frequency shift, is about 5% or less. Okay. This particular platform has, it's made of silicon. First of all, has a square or nearly square platform and 4 little springs. Okay? And let me show you a bigger picture of this. So these are the springs, and there's also this platform that you can plant cells on. And if you look at these lighter regions, this is coated by gold. So there's a circuit on this platform that you can have a current going through that particular platform. Okay. And the way that we measure the resonance frequency is when you have the current going through. Let's say you supply a cyclic current going through. And there's then the cyclic magnetic field surrounding that particular platform. If you have a permanent magnet, then because of the cyclic magnetic field, this thing would vibrate up and down. Okay, and for different frequency of the current, that vibrates at different frequency. Okay, and the vibrational amplitude is very limited. Unless you hit the resonance frequency, then the amplitude would start to grow. That's how you measure the resonance frequency or shift of the resonance frequency. Okay. All right, you can certainly culture cells. Not again. I actually tried this right before lunch. Give it a try. Huh. Let's see if this one works. Oh, somehow that's discouraging. Okay, let me just show you. Okay, so this is just the culturing of the cells on the platform. This is the vibrational platform, and the cells we used was HT29 NIH HT29 cells. This is a colon cancer cell, and we measured the cell frequency shift as a function of volume. It seems to be a straight line. So everything seems to be working okay. This is these cells cultured on the platform. That's an SEM image of this. Okay. Okay. Now, we can, the first thing we did was we want to calibrate this thing, okay? So we measure the cell frequency shift. From that, we obtain the so-called mass of the cell, and we want to see whether the mass we measured is a few nanograms and so on. We want to see the mass we measured is real, is something really related to the cell mass itself. One thing we did was we measure the cell mass, then we immediately fix the cell, kill the cell. Okay? Then after we kill the cell, we measure it again using the same platform. We kill it on the platform, then we measure it again. The argument is right before the cell died, And right after the cell died, the cell should not change its mass. Okay. This is the result we got. This is the apparent mass before fixation, before the cell died. This is the apparent mass after the cell died. Okay? And it's a straight line, but the slope is not 1. The slope is about 1.4. We were really puzzled. In other words, okay, let's say I'm about 130 some pounds. Okay? If I died right now, you measure me, I'm 200 pounds. This is impossible, right? So right before the cell died and right after the cell died, there seems to be an apparent increase in the cell mass. This is what the other people call ghost mass phenomenon. It's not, we're not the first one to discover this so-called ghost mass phenomenon, but we're the first one to explain that, I believe. Okay? And the reason for that is most of these models, mechanical models, to explain or to measure the cell mass is based on this particular mechanical system. You have, whether it's a cantilever beam or the platform we have, you have a system which has a mass, a spring, and dashboard, and so on. When you have a cell added onto it, you added an added mass, M2, into the system, and that's what you measured. Now the question is whether this is a good mechanical model to measure this particular cell mass or added mass. It turns out that it's not a very good model. Here are some finite element simulations that show that the cell you're measuring, the behavior of the cell strongly depends on the stiffness of the cell. Okay? If the stiffness is low, as the vibration platform vibrates, the cell deforms significantly. If the stiffness is high, the cell doesn't deform as much. That actually has a significant effect on the frequency. The resonance frequency is on the order of 16,000 hertz, 16 kilohertz. Okay, so what we did was we actually developed a model based on this model. Instead of assuming the cell as a dead mass, we assume the cell as a mass with a stiffness or with a spring and a dashboard. This again is a very crude model compared to the real situation. So the real situation, the cell is a body, right? It's deforming almost with an infinite degree of freedom. This is simplifying this entire system of the cellular system as one mass, a spring, and dashboard. Nevertheless, this is conceptually very different from that model. And based on that, we actually can calculate, indeed, for enough number of tests, you can calculate the effective stiffness of the cell, the effective mass of the cell, and the effective viscosity of the cell. Okay. And this is how we did that using maybe 16, 20 different tests. We got the effective stiffness of the cell before fixation to be roughly about 4 kilopascal, and after fixation about 7.9 kilopascal. Okay, so the answer to, or the result of that showed that the cell stiffens significantly if you fix it. Because of that, the response of the cell when you measure it would be quite different. Okay? Here are some measured results. This is the calibration. When there is a cell on it, you have a mass of less than 1 nanonewton. And if the cell falls off, then the mass drops. Okay? When the cell grows, the measured cell mass grows. Okay? And if the cell divides, actually divides from one cell to two daughter cells, we actually observe some kind of a dip over here. Let me show you, maybe it's a better way to do this by a movie. This is cell growth measured by our device. That's again cell growth, and dividing into two daughter cells. At the location of or instant of division, there seems to be a dip of apparent cell mass, okay, or measured cell mass. That's again cell growth and dividing into two daughter cells, a dip. Two daughter cells dividing into four daughter cells, another dip. Okay, consistently, very consistently, we observe that. Yes, Larry.
Quick question. So, can you excite a second mode when you have a second spring mass on there?
Yes, you can. Very good point. But what we did was we always took the first mode, the lowest resonance frequency for the combined system.
And does the vibration itself affect the behavior of these cells? So can it influence how they divide and how fast they divide, the vibration?
Okay. Our argument is it's not, because it's not. Okay, the way we do that is we grow the cells, culture the cells on the platform, and for about 20 minutes, half an hour, then we do a vibration to measure the cell mass. Then again, stop the vibration and let it grow and do a test. But even the right, we don't know. We don't know. So one way to test that is actually, let's say, you can do this every half an hour or so, measure it, or you can have another test that runs several hours without any vibration, then measure it to see whether you get consistent results. But I'm sure, as you know, for all these cellular studies, the scattering is fairly large. So from one measurement to another measurement, you don't always get very consistent results. But we hope that that's not changing. That's a good, we didn't do bone cells, but we could actually. Okay. Yeah. Very good. Very good. Yeah. Okay. Let me go back to the picture over here. What we found was exactly what you said. Right before the cell divide, they tend to ball up. Remember the picture, the bright spot in my movie? That's how the cells behave. Okay? Right before they divide, they tend to ball up. And then the mechanical system is different from a spread out cell and the balled up cell. Right? And because of that, you have an apparent dip of the measurement. I actually have a very simple mechanics model to explain that. I thought I had that. Oops, I apparently didn't include this. Okay, I probably put it into a different file. Anyway, the model you can imagine is, let's say you have a little cylinder on the platform, okay, and the center of mass is at the center of the cylinder, okay. With the same mass, if the cylinder is fatter and shorter, then the center of mass is lower. Okay? And because of that, the mechanical system is different for a slender cylinder versus for a fat cylinder. You can actually, using a very simple mechanics analysis, you can actually show that as the height of the cylinder goes to 0, the system goes to infinity. Okay? So if the cell balls up, then the apparent stiffness or equivalent stiffness actually decreases. That gives rise to the dip of the cell mass measurement using this particular technique.
Platform is uniform.
Yeah.
But during some, could there be a chemical gradient that change the medium when the assumption is not correct? Or there's some
The density change?
the local density change will affect the measurement?
In what sense? What is the mechanism that causes the density change?
Consumption of some of the materials in the medium or
Not very likely. I don't see any particular mechanism. The frequency, depending on the frequency range, may affect the so-called viscosity. Right? There's a certain range of velocity that the viscosity is linear, or the strain rate, stress versus strain rate curve is linear. Beyond that range, the nonlinearity may come in. But density, I don't see any good reason that should change due to vibration. Yeah. Okay. All right. So this is just to show that indeed we can measure the cell mass as a function of cell growth rate as a function of cell mass. And in this case, it appears to be somewhat linear related, or with a very slow power law. Okay. Oops. I think I must have put the remarks in the wrong file. Also, this was not updated, so I was working on two different files. The other file had the updated information, and this file doesn't. Anyway, these are the collaborators, and it's supported by National Science Foundation and Army Research Lab. But just to conclude, if you look at the biological processes, perhaps the most important, one of the most important processes is cell division. Let me see if I can find that picture or movie over there. Okay, this is the movie of cell division. This is a single cell, these are chromosomes. Right before the cell divide, some of you may have seen this movie before, and they line up. This is the process that the cell tend to ball up, then divide into two different daughter cells. It's a fascinating process. Actually, I downloaded it from the web. My sense is using technologies and collaboration between physicists, engineers, biologists, medical scientists. If one day we can solve that problem, that would be a huge advance for science and biology. Okay, with that, I can take some questions. No. Our cells are consistently fully adhere on the surface of the platform. We didn't observe any secretion of something out of the cell.
Can you also do some control experiments and you can fabricate different structures, same mass, in a smaller footprint, and a bigger
We did. We actually we made just polymer, little cylinders of polymer, and put it on the platform and measure it. That's for calibration. Okay. And we don't have the full results yet, but we certainly observe that different aspect ratio of this little added mass would give rise to different measurement results. We've done 4, maybe 8 at most for this particular type of cell. They will become pretty crowded quickly. You mean the distribution of the cells? There will be some error or some difference, but the difference would be somewhat limited because in our finite element simulation we show that if you move the added mass at any point on the platform, the error induced by that is less than 4.5%.
Cell number on the platform, and would that actually the difference of the cell mass? I mean, the colony and the cell.
If they become confluent, if they started to interact with each other, then it may change the measurement results. Or the question you may ask is, let's say instead of just one layer of cells, if you put several layers of cells, then you're changing the geometry of this added mass. The geometry of the added mass and the dynamical response of the added mass would affect the measurement results. Yes, Roger.
And then the viscoelasticity.
That's correct. Right. Yeah. Yeah. Okay. Thank you very much.